Giant cell lesions. Introduction and definition. History. Cell of origin of giant cell. Formation of giant cell. Concepts of fusion of giant cells. Molecular mechanism of fusion of giant cell. Classification of giant cells. Brief on each giant cell types. Classification of giant cell lesions. Chattopadhyay A in 1995 and Varghese I and Prakash in 2001. Brief on each individual giant cell lesion. Introduction to giant cells. Giant is an English word which was coined in 1297. Giant is an English word coined in 1297, which means beings which are very large. Beings which are very large when compared to normal. The American Heritage of Medical Dictionary defines giant cell as an unusually large cell, especially a large multinucleated phagocytic cell. Giant cell lesions are a group of lesions in which giant cells are found. Presence of giant cell is often pathognomonic and provides vital clues in the diagnosis of various oral lesions. History. Muller J. in 1838 discovered giant cells. Muller J. in 1838 discovered giant cells. Virchow and Langhans discussed their nature. Langhans et al. First reported multinucleated giant cells in tuberculous granuloma. Langhans et al. First reported multinucleated giant cells in tuberculous granulomas. The cell of origin of giant cells. By Saurabh R. et al. Giant cells in overview 2020. He divided cell of origin of giant cell into 5 categories. Proliferating giant cells associated with tooth eruption. Endothelial cells. Proliferating mononuclear cells, monocytes, and mitotic or amitotic division of monocytic nuclei. Coming to formation of giant cells, how giant cells are formed. This was given by Faulkner in 1930. He explained 2 theories by experiment. One was nuclear division of monocytes with the absence of cytoplasmic division, and the other was fusion between monocytes. So, in the nuclear division of monocytes with the absence of cytoplasmic division, there is a monocyte and it divides. Only the nuclear divides and there is no cytoplasmic division. And then it keeps on repeating. Because of this, the nucleus from here, they kept on repeating towards the periphery and a rosette pattern of nuclear arrangement is seen. This is seen in Langhans type of giant cell. It was given by Shrija et al. A review on giant cells of oral cavity 2021. Now what happens in fusion between monocytes? 2 monocytes or more than that, they fuse multiple monocytes, and then because they are fusing, so irregular arrangement of nuclei in the central to like the all over foreign body type of giant cell. Coming to concepts of fusion of giant cells. Immune-mediated concept. Lymphokines and cell surface changes facilitate the fusion of macrophage. In lymphokines and cell surface changes facilitate fusion of macrophages. Young and old macrophage concept. All macrophages has chromosomal abnormality and altered cell surface. This is recognized by a young macrophage, and then there is fusion of cells. Now there is something called as endocytic activity concept. 2 or more macrophages, they try to ingest or engulf the same antigen. If there is one antigen and 2 or 3 macrophages are trying to engulf it, it results in the phagocytosis and then the fusion of endosome margins between the 2 cells, and therefore there is a multinucleate giant cell. And there is something called as induction by viruses. The viral antigens gets incorporated into a polycryon cell membrane and thus multiple cells. Then reduction in the cell coat thickness forms a bridge between 2 cells. Cells enlarge into a complete cell fusion. Coming to molecular mechanism of fusion of cell. So here In the cell, we have a cell. There is a nucleus. On the cell surface, cell membrane, we have E-cadherin as a part of the cell membrane structure, and we have an antigen outside called CCL2. So it's a chemotaxis. It will do chemotaxis. So this is a cell. There is DAP12 and STAT6. DAP12 and STAT6 induce a fusion competent status. Chemotaxis of macrophages towards each other is mediated by CCL2, and cell-cell adhesion is mediated by E-cadherin. Substrate adhesion is mediated by beta-1 and beta-2 integrins, and then cytoskeletal rearrangement is mediated by RSE1. Cytoskeletal is rearranged. There is lipid recognition, there is phosphatidyl serine exposure, and exposure is mediated by CD36. Exposure of phosphatidyl serine and lipid recognition is mediated by CD36, and this leads to the fusion of cell. This entire was given by Helming et al. Molecular Mediators of Macrophage Fusion in 2009. Then coming to the classic types of giant cell. Giant cells can be physiologic or pathologic. Physiologic giant cells are osteoclast, megakaryocytes, trophoblasts. And pathologic giant cells are giant cells in inflammation and giant cells in tumor. Giant cells in inflammation are foreign body giant cell, Langhans giant cell, two-tone giant cell. Giant cells in tumor are tumor giant cell and Reed-Sternberg giant cell. This is given by R.S. Gupta et al. Giant cells in health and disease in the year 2016. So coming to the physiologic type of giant cells, we have osteoclast. Number one is osteoclast. This is how an osteoclast will look like. It morphologically resembles a foreign body giant cell. The nuclei is about 10 to 20 in number. The origin is mononuclear cells of hematopoietic stem cell, and the role is to bone remodeling and calcium hemostasis. Coming to megakaryocytes. Megakaryocytes are large polypoidal cells, multinucleated or multi-lobed nucleus. Eosinophilic granules are there, these purplish granules, and the origin is hematopoietic stem cell again. The role is a platelet precursor. Coming to trophoblast. Trophoblast is the first cell to differentiate from a fertilized egg. The role is in implantation of the embryo, exchange of nutrients, waste and gases between maternal and fetal system. Then we come to pathologic giant cells. The first is foreign body giant cell. This is a foreign body giant cell. It is formed by the fusion of macrophages M2A. The structure is up to 100 to 200 nuclei with a uniform size and shape, found at the tissue material interface, because it is foreign body, right? So some kind of material and tissue will be there. So it is found at the interface of tissue material interface of the implanted medical devices, and found in foreign body granulomas. The role is foreign body response, degradation, resorption of chronic inflammation. It is positive for CD68, E-cadherin and DC stamp. Then comes Langhans giant cell. Langhans giant cell could either look like this or this. There are 15 to 20 nuclei, which can be arranged either at the periphery or at two poles. The appearance is horseshoe shape or cluster at two poles. It was first described by Langhans, that's why it is called Langhans giant cell. Commonly seen in granulomatous infections such as tuberculosis, leprosy, late syphilis, deep fungal infection, sarcoidosis and orofacial granulomatosis. This is positive for CD68 and CD40. Then we come to two-tones giant cell. Two-tones giant cell is also known as xanthelasmatic giant cell. So peripheral, here we see foamy vacuolated cytoplasm, and the center nuclei is arranged in a wreath-like ring-like or wreath-like. Associated with xanthoma, fat necrosis, dermatofibroma, fibrocystocytoma and xanthogranulomas. Positive for CD68, factor 12A, lysozyme and alpha-antitrypsin. Then comes Ashkoff giant cell. In Ashkoff giant cell, there are about 4 to 5 nuclei, which has a characteristic appearance of caterpillar nuclei. It is a cardiac myocyte, originates from cardiac myocyte and associated with rheumatic heart disease. Then comes the class Reed-Sternberg cell. It has a very classic owl's eye appearance, which is a bilobed nucleus. There are variants of Reed-Sternberg cell, which is seen in Hodgkin's lymphoma mostly. They are coin on a plate appearance, mummified cells, popcorn cells, and lacunar cells. The formation of Reed-Sternberg cell is from B cells of germinal center. It is positive for CD16 and CD30. The diseases associated are pathognomonic of Hodgkin's lymphoma, but it can also be seen in infectious mononucleosis and other lymphomas. Comes two-toned giant cell. Tumor giant cell. Tumor giant cell means it forms when the tumor cell possess abnormal surface. So they produce extracellular enzymes. These reduce the surface coat thickness and approximates the bilobed layer, and there causes a fusion. This is positive for MIB1. The disease associated is giant cell tumor of bone. So how it looks like? Polymorphic nucleus usually diploid or like this also, and hyperchromatic nucleus shows abnormal mitosis. Then comes Zank giant cells. Zank giant cells. The structure is bizarre cells, atypical in shape, and nucleus is crowded with peripheral margin of chromatin condensation. Formation epidermal in origin, viral altered cells. Disease associated is herpes simplex, varicella zoster, and cytomegalovirus. Then comes melanocyte-derived starburst giant cell. Melanocyte-derived starburst giant cell. Here we can also see pigments. The structure is a multinucleated melanocyte which appears stellate because of a prominent dendritic process. The disease associated is lentigo maligna. Then comes Warthin-Finkeldy giant cell. Warthin-Finkeldy giant cell. The formation, it can be through live virus or inactive virus. Live viruses penetrate the cell, form viral coded protein, and cause fusion of the cell. But in inactivated virus, they fuse to cell surface and reduce cell coat thickness, and cells come close, and also fusion is there. So we have faint cytoplasmic border, and there is cluster of nucleus which is grape or mulberry like, about 3 to 30 nuclei. Then comes nevus balloon giant cell. Nevus balloon giant cell. The structure is clear vacuolated melanin granules cytoplasm. The clear vacuolated we also saw before in two-toned giant cell. In nevus balloon giant cell also clear vacuolated melanin granule cytoplasm. Here formation is by union of degraded melanosomes. Disease associated is blue nevus, malignant melanoma, melanocytic tumor of eye. Then comes the classification of giant cells. This was given by Chattopadhyay A in 1995. Based on the etiopathogenesis with pathognomonic and non-pathognomonic association. Lesions where giant cells are pathognomonic. Giant cell fibroma, peripheral giant cell granuloma, giant cell tumors, and Hodgkin's disease. Then number 2, lesions where giant cell is characteristic but not pathognomonic, such as tuberculosis, HSV infection, measles, xanthoma, and cherubism. Then lesions associated with presence of giant cells, such as orofacial granulomas. Orofacial granulomas, fungal infection of foreign body reaction, neoplasms, syphilis, leprosy, fibrous dysplasia, Paget's disease of bone, aneurysmal bone cyst, ossifying fibroma, Wegener's granulomatosis, actinomycosis, chronic diffuse sclerosing osteomyelitis, odontogenic giant cell fibromatosis, Heer-Frost syndrome. Then comes based on the etiopathogenesis. This was given by Varghese Prakash in 2011. Varghese and Prakash in 2011. Here we divided on the basis of the etiogenesis. Microbial lesion, tumor-like lesion, cystic lesion, metabolic lesion, osteodystrophic lesions, and miscellaneous lesions. So coming to microbial lesions. The microbial lesions are tuberculosis, leprosy, actinomycosis, and sarcoidosis. The tumor and tumor-like lesions are central giant cell granuloma, peripheral giant cell granuloma, giant cell fibroma, giant cell tumor, osteosarcoma, rhabdomyosarcoma, Hodgkin's lymphoma. Cystic lesions are aneurysmal bone cyst and traumatic bone cyst. Aneurysmal bone cyst and traumatic bone cyst. Metabolic lesions are hyperparathyroidism, osteodystrophic lesions are Nuna-like multiple giant cell syndrome, Nuna-like multiple giant cell lesion syndrome, and miscellaneous such as cherubism, Paget's disease, or fibrous dysplasia. This much is till the classification. In the next video, we will cover each lesions in brief.